Today we are going to start our new unit, that is genetics. Two chapters come in this unit. The first is Principle of Inheritance. And the second is the molecular basis of inheritance. The third chapter, that is evolution, has been completely deleted in the revised syllabus of 2020. Now these two chapters, Principle of Inheritance and Molecular Basis of Inheritance, are of 18 marks. Both these chapters come under genetics. So, the study of genes is known as genetics, and Gregor Johann Mendel is known as the father of genetics. Gregor Mendel's work till today is considered, appreciated and found to be correct. Gregor Johann Mendel was born in the year 1822 in Austria. Mendel was the first person to give about heredity, to tell about heredity. But his work went unnoticed and unappreciated till 1900. In the year 1900, some biologists like Hugo de Veres, Karl Koren, and Schamak worked and studied his whatever theory he gave and found that whatever Mendel gave was correct. Now, if we have to understand Mendel's work and study genetics, we have to be familiar with certain terminologies that are used in genetics. So, let us know about those terminologies. First is factor. When Mendel was working on genetics, he did not know the term genes. Genes was not coined. The word genes was not coined at that time. So he used certain substances. He knew that there are certain substances or particles that are transmitted from one generation to another generation. So he called those substances as factors. But today we know that these factors are called as genes. So these factors are known as genes. The term genes was coined by Johansen. The word genes was coined by Johansen. Genes are the word gene, which was coined by Johansen, They are units of heredity which has the information and it controls the characters. It has information that can be transmitted from one generation to another generation. So genes are found in the DNA. You can say genes are the segments of DNA. The next term is allele. Alleles are the alternative forms of a gene. Normally a gene has two allelic forms. If it has got more than two forms, allelic forms, then it is called as multiple allele. Now we come to phenotype. Phenotype is the observable morphological character which is visible. So that is known as phenotype. So this word phenotype was also coined by Johansen. The next term is genotype. It refers to the genetic constitution of an organism for a particular character. The word genotype was also coined by Johansen. Next term is homozygous. It refers to a condition. Homozygous, it refers to a condition in a diploid cell where two characters are similar. So, like capital T and capital D, that is tall, or small t and small t, that is dwarf. Heterozygous, it refers to a condition when two alleles of a character are not similar. Then it is known as heterozygous, like capital T and small t. The next term that is used is cross or hybridization. It is an act of sexual reproduction that is carried on between two desired organisms. Though it occurs naturally, it can be made artificially, it can be used artificially for experimental purpose. In the study of genetics, we come across many types of crosses like monohybrid cross, dihybrid cross, then test cross, back cross. So the next cross is monohybrid cross. In this cross, only one character is considered at a time for analysis. Suppose we are taking height of the plant. So if we take only height, then it is a monohybrid cross. Then the second is dihybrid cross. In this cross, in the dihybrid cross, when two characters are taken simultaneously for an analysis, then it is called as dihybrid cross. Suppose we are taking the height of the plant and the shape of the seed also, then it will be a dihybrid cross. Now we come to back cross. If a cross is made between the parent and the progeny, then it is known as back cross. Back cross involves the cross of the F1 progeny with either of the two parents. Now we come to test cross. Test cross is also a type of back cross, which involves the cross between F1 progeny with homozygous recessive parent. The test cross is used to find out the homozygous or heterozygous nature of the F1 progeny. F1 and F2 generation. F1 means the first filial generation. This is F1 is first filial generation. When a fresh cross is made, then it is called as F1 generation or first filial generation. The progeny obtained from a fresh cross is known as F1 generation, and F2 generation means a cross made, that is selfing. Selfing means a cross between the male and female between the F1 generation, that is the progenies of the F1 generation is called as F2 generation.